All right, before we go on and we think about how this whole process gets mapped on with the metabolism of other fuels like lipids or proteins, let's take a little bit of a metabolic bookkeeping, right? Let's take stock of sort of where we've been. So I love this picture too. It's a little bit of a simpler picture than we just sort of had, but reminding us of the processes that we've been through. So again, we're going to distinguish where we're doing biochemistry, whether we're in the cytoplasm or the mitochondrion. I'm going to highlight that sometimes we will use this term cytosol. There is technically a difference between cytoplasm and cytosol. I'll tell you what it is and then indicate that you may hear these terms interchangeably. So when you refer to cytoplasm, that is essentially the liquid part of the cell with the organelles. And then cytosol is just the liquid part. So as a better way maybe to think of it with an analogy, if we think about soup, right? Soup is really cytoplasm. It has the broth plus the pieces in the soup. Cytosol is just the broth, okay? I generally refer to the term cytosol and cytosolic and mitochondrion and mitochondrial, but just wanted to make sure you understand if I use those terms interchangeably or if you see them to understand that cytoplasm and cytosol are generally referring to the same metabolic compartment. So if we think about the processes that we've talked about that are cytosolic, while we will learn many more, we've sort of just centered ourselves on thinking about glycolysis, right? And glycolysis is this process that's over there, glucose to pyruvate. Now, when we think about the words that we use to describe glycolysis, we said that it was rapid, but inefficient. And here's what we mean, right? It's rapid in terms of its ability to quickly make ATP. And it does so because it makes ATP by that process called substrate level phosphorylation. So again, that's a very rapid way to make ATP. You're just finding somebody that is a phosphorylated compound that's worth more than 30.5 kilojoules per mole, and ADP says, hey, give me your phosphate. I need to be remade into ATP quickly. They trade, and you quickly regenerated ATP. So substrate level phosphorylation is very rapid, but it's also inefficient. And remember, by that inefficiency, we meant that the end product of glycolysis was pyruvate, and we still had a lot of energy on the table with that molecule. In our sponge analogy, we've essentially just cut the sponge in half and just squeezed a little bit of water out. So pyruvate still has a lot of metabolic value to it. Other pieces just to highlight, we did generate some alternative currency that we have here, right? But glycolysis is a rapid but inefficient mechanism to make energy. So dipping into the mitochondria, and remember, we said that we really didn't dip into the mitochondria unless you had oxygen around. So thinking about sort of where we're at here, right? Once we dip into the mitochondria, we say that we're mitochondrial in terms of the processes, mitochondrial. If we think about these steps that happen there, again, we are going to say that we don't sort of step in there unless we have oxygen around, unless we are aerobic. Okay, so we have pyruvate dehydrogenase chemistry that happens there. So here's the pyruvate dehydrogenase chemistry, right? The conversion of pyruvate to acetyl-CoA. We liberate CO2 and we generate some of those reduced cofactors. So here's some more of that NADH. We've taken our fuel molecules and oxidized those carbon atoms fully to CO2. Okay. Another thing that we talked about that happens in the mitochondria is we've got the citric acid cycle. Right? So again, here is the citric acid cycle. Okay, and I should highlight, I said up here that we have the bulk of the CO2. We don't have the bulk of the CO2 left at this point. We do snip off some of the carbon atoms of CO2 as CO2, but the bulk of the carbon dioxide actually comes off in the citric acid cycle. Again, the citric acid cycle allows us to make some alternative currencies that we harvest and take onto the electron transport chain, as well as some primary currency. Noting here that it really does synthesize GTP, but that is net energy equivalent to ATP. All right, and then the last thing is electron transport and oxidative phosphorylation, right? Our fourth step here, we are going to cash in all of these sort of reduced cofactors. We're going to generate a protein gradient, use that protein gradient as a way to synthesize ATP. So if we highlight some of the pieces that we talked about with our mitochondrial processes, these three things that happen here, we say that these are much slower, so these do not produce energy rapidly, but importantly, they are very energy efficient. We squeeze, and let's maybe say that we're going to say use two colors here. They're energy efficient because if I use these two colors here, I'm going to highlight that all of the carbon atoms they're fully squeezed bone dry, right? We've cut them all apart and they're liberated as CO2, fully oxidized and efficient. We have harvested all of that water from the sponge, electrons from our fuels, and we've stored them either as these reduced cofactors, which we're going to cache in the electron transport chain, or we've directly made some ATP, both a little bit in the citric acid cycle and then in that anaerobic process of glycolysis. So maybe a simpler 30,000 foot view that we have here, but here's some important pieces that I do want you to think about. I want you to know where different things are made. These are some of the questions that you're likely to see on quizzes or exams, is being able to think about where these processes or where these products are made. So know where they are made. And remember, here's our fuel equation, right? Fuel plus oxygen generates carbon dioxide, water, and energy. And when we think about energy, we're going to think about it in two forms, right? And I'm going to color this kind of as green for our primary energy and then orange for our alternative currencies. So when we think about metabolites, right, fuel is ultimately converted all the way into CO2. So I want you to know where CO2 is made. Now, we're going to learn lots more places next semester when we go and take a deeper dive on many of these metabolic pathways. But for right now, we know that pyruvate dehydrogenase chemistry is going to liberate CO2 and the citric acid cycle. So again, for right now, I want you to know where these pieces are made. When we think about specifically the energy as ATP, we know that ATP can be made in one of two ways. We can make it by substrate level phosphorylation, and we can make it by oxidative phosphorylation. So I want you to know sort of where those processes happen. So again, we've got our primary energy, substrate level phosphorylation, or oxidative phosphorylation. Now, we're going to see lots of instances of substrate level phosphorylation next semester, but right now we know that that happens with glycolysis and that happens with the citric acid cycle, specifically when we're making GTP. Oxidative phosphorylation, well, that's because it's part of that electron transport oxidative phosphorylation process. So that's where we're going to make ATP. When we think about those alternative currencies, we want to think about NADH, okay? So again, one of the alternative currencies here is NADH. We need to know that that is made in glycolysis, pyruvate dehydrogenase, and the citric acid cycle. We make that all over the place. So we've got glycolysis, the citric acid cycle. Oops, let's put pyruvate dehydrogenase. Let's keep these in order here. Pyruvate dehydrogenase chemistry, and then the citric acid cycle, and then FADH2. Again, we will learn more places where we find these, but for right now, the only place that we've seen FADH2 is the citric acid cycle. Okay? Bottom line, this whole process nets 32 ATP. Okay, so some metabolic bookkeeping that'll allow us to keep track of these 4 steps that we've talked about for the full metabolism of glucose.